Hello students, welcome to EPG Pathsala. My name is Anant Narayan Bhatt and I am working as a scientist in DRDO Lab, Institute of Nuclear Medicine and Allied Sciences. So today we are going to talk about enzyme regulation and feedback control from the paper Molecular Enzymology and Protein Engineering in Biophysics. So in this chapter, we intend to learn about the following aspects of enzyme activity. These are mechanisms of control of enzyme activity. And what are these mechanisms? They are the substrate and product concentration, then the turnover of protein, like the synthesis and degradation of the protein whenever it is required, and then by expressing various forms of same enzyme, like which are called isozymes, like hexokinase, which have 4 isozymes sometimes in the same cells or sometimes in the different tissue wherever physiologically it is required. So sometimes these isozymes they express in different type of tissues. So this is another mechanism. Then we will be studying about the reversible modification, which is a reversible post-translation modification, which is also a mode of regulating the enzyme activity. Then proteolytic control is one mode of mechanism of regulating the enzyme activity. Then allosteric control, a majority of the metabolic enzymes, they are basically regulated through allosteric control for their enzymatic activity. Then how sometimes the substrate or the intermediates formed in any pathway, sometimes they can also regulate the activity of enzyme through feedback inhibition. So we'll be talking about feedback inhibition. Then we will be studying about the kinetic behavior of allosteric enzymes, which includes the competitive and non-competitive inhibition. Then we will be studying about the mechanism of allosteric interactions. So in cells, at any given time, there are thousands and thousands of reactions are continuously going on. So at any given time, there's so many reactions which are going on. If there is no control or if there is no proper regulation of the enzyme activity or the reactions occurs, then what will happen? There will be havoc. So like cell is a very nicely designed unit which can regulate each and every chemical reactions through basically fantastically evolved and designed enzymes. And how it can regulate, because the enzymes have fantastic regulation for their reactions. They have very nice specificity, three-dimensional specificity, so that they can bind very specifically to their own substrate only, so that the reaction can occur in very nice way. So the activity of protein enzymes must be regulated so that the function of the cell can be easily achieved in proper time, because time is also essentially required. So it can be achieved in proper time and at proper place. So the control of enzyme activity is essential in maintaining the steady state for any cellular organism, because if the steady state is not maintained, the E. coli, which can easily divide and form two cells in 20 minutes. So if the enzymes are not performing perfectly, then the E. coli cannot divide and form reproduce continuously. So from that perspective, each and every enzyme should work in very proper and controlled manner. And then the functional expression of all these enzymes appears and disappears depending on the cellular need. So at any given time, what is happening, like there is limited resource. So if some reactions is not required, the enzyme of those enzymes will not be present. They will be degraded and the basic unit will be used to synthesize the new enzyme. So and all these biochemical pathways which can take place in the cell does not operate in full swing at any given time. So rather, so that is how, some enzymes are stopped, some enzymes are degraded to form the new enzymes. So that's how the biochemical pathways are regulated in very controlled manner in any given cell to give optimal function and maintain the homeostasis, cellular homeostasis. So this explains a very nice way how the physiology basically regulates the enzymatic activity inside the cell. So the physiology means what? Like when there is more glucose is there, when there is high blood glucose, the situation A you can see, when there is the concentration of the blood glucose is very high in the cells, what is having the circulating glucose levels are very high. So enzymes are controlled in such a way that at high glucose concentration, liver store the glucose in the form of glycogen. So how it occurs, like which will be talking more in detail in coming slides. Like the liver has a enzyme which is called glucokinase, which has high Km value for glucose. So high Km value means it has low affinity for the glucose. So whenever there will be more glucose in the blood, then what will happen, like the glucokinase will be activated, and then glucokinase will push the glucose more towards glycogen synthesis. So the glucose will be stored in the liver for any time when there will be a requirement of energy, and the glycogen can break down, and then it can produce more glucose, and then it will again come back into the bloodstream. So there is another scenario B, where the blood glucose is low and body needs the glucose for making energy from the liver cells. So the glucose is already stored in the liver in the form of glycogen, and the liver cells activates another biochemical pathway to break down the glycogen, and that biochemical pathway is called glycogenolysis. So based on the physiology, based on the requirement of physiology, when the blood glucose is high in the circulation, the another pathway gets activated which is called glycogen synthesis, and there are set of enzymes which can convert the glucose in the form of glycogen in scenario A. And there is another physiological requirement when there is low blood glucose in the bloodstream. Another set of enzymes gets activated, and they are nicely controlled and regulated, and then they break down the glycogen through glycogenolysis, and then they produce more and more glucose which enters into the blood and fulfills the requirement of physiology of more and more glucose in the blood for synthesis of energy. So we have seen like how nicely the enzymes are controlled and regulated. The biological activity of enzymes and proteins, they are regulated in many ways. I mean, there are several ways by which the biological activity of protein and enzymes can be regulated. So the very first method or regulation type of regulation is the substrate and product concentration, then protein turnover. We will be discussing about all these types in detail in coming slides. And then the next is by expressing various forms of enzymes. So isozymes are those various forms. They have different Km values, and at different level of substrate concentration they act in different, and they sometimes they express in different tissue types also. So based on the requirement, they express in different tissue types, and then they act differently. Then the next method of regulation of enzymes is reversible post-translational modifications. So sometimes what happens, like some moiety gets covalently attached to the protein, and that brings a structural change, and it can switch on or switch off the enzyme molecules, and then that's how it regulates the protein. Then the next is proteolytic control. Sometimes what happens, like some enzymes, basically some proteases, if they will express in active form in the tissue, in any cell or tissue, they will basically cleave, or they will proteolyze their own tissue of origin. So they generally express in inactive form or precursor form, and then they get activated at the time of at the site of action by some proteolytic mechanism. That is another way of regulation of enzyme. Then there is allosteric control, which is in which some substrate molecule can regulate the binding or inhibition of some enzyme molecules. So these are different way how the enzymes or proteins, they can regulate their biochemical reactions. So when we talk about enzyme regulation, it is basically about the protein activity or the enzyme activity. So the activity of proteins or enzyme must be regulated so that they function at the proper time and place. So the control of enzyme activity is essential in maintaining the steady state of all organisms, and that is why it should occur in proper time and place. So the functional expression of particular set of enzyme appears and disappears depending on the cellular need. So therefore all the biochemical pathways which can takes place in the cells does not operate in full swing at any given time. Rather, these pathways consisting of enzymatic reactions are regulated in very controlled manner to give optimal function and maintain the homeostasis in the cellular or the physiology. So, for example, glycogen synthesis and breakdown, both are processes occur in liver cells, and they are tightly regulated and work exactly opposite to each other. So, and this depends on the physiological requirement. So, depending on this physiological requirement, the systemic signals starts glycogen synthesis in liver cells. However, when body runs out of energy source, that is glucose, it sends a signal to liver cells for the degradation of stored glycogen, which was stored when the glucose concentration was in blood was high. So therefore, both these biochemical processes which acts against each other can take place in same cells but not at the same point of time. So that is why the enzyme regulation is essentially required. So the disruption of this highly ordered enzymatic control can disturb the homeostasis and can lead to severe pathological conditions. So we will talk about regulation of enzyme activity by protein turnover in this section. What happens basically, many a times what happens in cells, the substrate concentration goes very high. So what does it indicates? That if there is substrate concentration is very high, that means the reaction is not occurring. So what will happen, that the cell has a cell signaling and regulatory mechanism in such a way that these substrate molecules, many a times what happens, that they can induce the expression of their target proteins. So what will happen, like it may bind to some transcription factor, or somehow it will increase the expression of those proteins. So amino acids will the protein synthesis will occur from amino acids, and this can be directly influenced by substrate concentration. So substrate concentration will increase the constant of synthesis, and then there will be more and more protein synthesis. So for example, HMG-CoA reductase and cytochrome P450 are the two enzymes which are inducible, which are known as, which is inducible. So whenever their substrate concentration will go high, there will be more synthesis of there will be more synthesis of these enzymes. So similarly, what happens, like if the substrate concentration goes down, that means these enzymes are not required. So in that case, what will happen, the degradation constant will go high. The product, sometimes the product itself influence the degradation in such a way that whenever there will be more product accumulation, there will be no need of the enzyme at that point of time in the cell. So there will be more and more degradation of the enzyme will take place in that scenario. So that's how the enzyme regulation can occur through protein turnover. In this section, we will talk about how the enzyme regulation takes place through the regulation through the isozymes. So isozymes are basically the enzymes which are basically two different proteins, but they are catalyzing the same reaction. So the isozymes provide an avenue for varying type of regulation of the same reaction at distinct locations or times. So isozymes are homologous enzymes within a single organism that catalyze the same reaction but differ slightly in structure and more obviously in Km and Vmax values. So their Km is different, so that means sometimes in different tissues their requirement is at differential glucose concentration, as we discussed about the glucose in liver and glucose in the muscles. So the beautiful example of this are hexokinase and glucokinase. So glucokinase is a enzyme which is performing the reaction converting glucose to glucose 6-phosphate in the liver for glycogen synthesis. However, in muscle tissues, the isozyme hexokinase is converting the glucose to glucose 6-phosphate and putting it for the more and more energy generation. So this is one example, and the other example is in different phase of life. So in different developmental phase of life, the hemoglobin and lactate dehydrogenase expression varies. So hemoglobin, basically the fetal hemoglobin, which has very high affinity for oxygen, is expressed. However, when the fetus is born, it turns to adult hemoglobin, it turns to human hemoglobin, which has relatively less oxygen affinity as compared to fetal hemoglobin. So as we discussed earlier, that in muscle cells, the requirement is in such a way that even at very low concentration, the muscles can make more and more energy. So the Km value of hexokinase 1 is very low, so that it can bind the glucose at even very low glucose concentration and can convert it into energy. So you can see in the graph that at very low glucose concentration, it is the reaction velocity is very high. However, in when there is not much requirement in the body of glucose in the body, what happens, like glucokinase gets activated and then it converts the glucose into the glycogen. So regulation of enzyme activity by substrate and product concentration. So how the substrate and product concentration regulate the activity of enzyme. So the rate of reaction of most of the enzymes are responsive to changes in substrate concentration, because the intracellular label of many substrate, I mean it can be any metabolic substrate, is in the range of Km value of the enzyme. So whenever the concentration will match the Km value, it will immediately bind to the enzyme molecule and it will the reaction will occur. So that's how the substrate plays an important role in the regulation of the substrate concentration plays an important role in the regulation of the enzyme. So if the concentration, if more concentration of substrate ensures more collision. So this is also another phenomena, like if the substrate concentration will be more, the chances of the collision will be more, and if there will be more collision, then this will lead to more enzyme and substrate complex. Thus, there will be an increase in the substrate concentration prompts an increase in reaction rate, which tends to return the concentration of substrate towards normal whenever there will be more enzyme substrate complex will take place, there will be more product conversion. So once the substrate concentration will go down, then what will happen, that then again the reaction will automatically stop. So the best example of the feedback inhibition, the best example can be of the feedback inhibition can be like the first reaction of glycolysis carried out by hexokinase is very good example of it. Like hexokinase, what it does, like it converts glucose to glucose 6-phosphate. So if glucose 6-phosphate accumulates in the cell, that means it is not being utilized by subsequent enzymes of the same pathway. So the glucose 6-phosphate will start accumulating, and this glucose 6-phosphate will bind on the allosteric side, where it will inhibit the where it will inhibit the binding of glucose to the substrate molecule on the active site. So it can act as a very good feedback inhibitor, and this mechanism, or this mechanism of enzyme regulation is called negative feedback control. The regulation can occur through covalent modification of proteins also, which are generally called post-translational modification in proteins. So inside the cell, enzymes are highly regulated through this post-translational modification, and one of the very common post-translational modification is the phosphorylation of proteins. The phosphorylation of protein can occur, or enzyme can occur for many reasons, but one of the major thing which gets regulated by the phosphorylation dephosphorylation is the switch on and switch off of the enzymes. So sometimes, let's say, like in this figure, you can see that the enzyme on the left side is it can be inactive or active depending on the enzyme type, and once it gets phosphorylated in the presence of kinase through ATP, what basically like if once it gets phosphorylated, if it was inactive, then it will become active, and either in some cases, if it was active in dephosphorylated form, after phosphorylation it can become inactive. And this activation inactivation is a reversible process. So whenever the purpose of reaction is enzymatic reaction is achieved, what will happen? The phosphatases will get activated, and those phosphatases will dephosphorylate this enzyme. So if the enzyme was active, this enzyme will become inactive, and then if the enzyme was inactive, because of dephosphorylation, this enzyme can become again active. So this is a reversible process which keeps going on, and in this case the enzyme is not being degraded. It is basically what is happening, that it is continuously maintained there, and the regulation is achieved by phosphorylation dephosphorylation. And these phosphorylation dephosphorylation generally occurs at serine, threonine and tyrosine residues of the protein molecule. So in many cases, when this phosphorylation occurs, basically at physiological pH, what basically happens that the phospho group has very high negative charge. So the propensity of the salt bridge formation with arginine or lysine residues is very high if this phosphorylation occurs at any site in the protein molecule. So what basically this is doing, that this change, this phosphorylation is bringing a structural change. So this structural change sometimes can make the proteins active, or sometimes they can make it inactive depending on what protein it is. So the phosphorylation induced conformational change influences the enzyme's intrinsic catalytic efficiency or other properties also, sometimes like the signaling properties which occurs, like in case of DNA repair proteins, what basically happens, like the phosphorylation of the DNA repair molecules basically phosphorylate others, so it activates the kinase cascade, and then it phosphorylates other molecules, then other molecules. So the chain of phosphorylation reaction starts. So it can do multiple functions. The phosphorylation of protein enzyme molecules can do multiple functions. So majority of cell signaling and metabolic reactions are predominantly controlled by phosphorylation and acetylation modifications in the cell. So another mode of regulation of enzyme activity is by reversible covalent modification. So naturally inside the cell, enzymes after synthesis, they can be easily modified on various amino acid residues. So they all are which are reactive, they can be easily modified. So what are these modifications? These modifications are called post-translational modification because they occur after the translation of the protein molecules. So these post-translational modification can be either phosphorylation, they can be acetylation, they can be methylation, they can be sulfonation, glycosylation, they can be amidation, they can be hydroxylation, they can be myristoylation, and so on. So and these modifications, most of the time it occurs in reversible fashion. So this is a very good method of regulation. Like phosphorylation, when phosphorylation of some enzyme occurs, what it does, like it it activates or it deactivates the enzyme. So there are some enzymes, when it gets phosphorylated, it becomes active. Similarly, there are some enzymes which, when it gets phosphorylated, it becomes inactive. So this is a mode of activation, switching on and switching off the enzyme activity. So phosphorylation and dephosphorylation permits the functional properties of the affected enzyme to be altered only for as long as it is serves a specific need. So once the need has passed, the enzyme can be converted back to its original form. Like the phosphorylation occurs in the presence of kinases, so enzyme gets phosphorylated. Similarly, to bring it back to dephosphorylated form, there are some phosphatases in the cells. So these phosphatases gets activated once the job is done. When the enzyme reaction is done, the phosphatases gets activated and they dephosphorylate these enzymes, and they become inactive or they become active. So that's how the covalent modification or post-translational modification plays an important role in regulating the enzymatic activity. So there is another mode of regulation of enzymatic activity, and that is by specific proteolytic digestion of some of the proteins. So certain enzymes are synthesized as proenzymes. So they are also called zymogens. So which are always expressed in inactive form of enzymes that become active only after being cleaved at a specific site in the polypeptide chain by specific proteases. So this regulatory mechanism generates digestive enzymes such as chymotrypsin, trypsin, and pepsin. So all these enzymes, they express as in the form of proenzyme, and once it is required, their activity is not always required. So whenever there will be requirement of their activity, they will be activated. From proenzyme, they will get converted to enzyme, and then they will act. So similarly, caspases, which are basically involved in programmed cell death or apoptosis, so they are also proteolytically get activated from procaspases to caspases, and then they execute the process of apoptosis. Then the process of blood clotting, it is so important. Like if these enzymes will express in the form of in active form, then blood will clot, it will not flow in our veins. So blood clotting is only required when there will be injury. So when there will be injury, these proenzymes, they gets converted into active enzyme, and then the blood clotting occurs. So because cell lack the ability to reunite, the enzymes, once this enzyme is digested and form two different parts, they cannot be reunited. Cell don't have any mechanism to reunite them. So this process of modification through proteolytic degradation is actually irreversible modification. It is not reversible. And then this is a very unique and specific mode of regulation occurs in the cell, and if this not occur, probably these proteolytic enzymes, they will chew the tissue of origin itself. So that is why this way of regulation of enzyme, proteolytic enzyme, is very essentially required in cellular systems. So we will be talking about regulation of enzyme activity through allosteric control. Allosteric control is very important control in regulating majority of the biochemical reactions. So allosteric enzymes are basically those enzymes which have a additional allosteric site beside the active site in the enzyme. So this allosteric site is basically the site where the effector molecule can come and bind, and that can regulate the catalysis of the enzyme. So Jacques Monod was the first person who proposed the existence of allosteric site in the enzymes which can regulate the biochemical reactions, and this site is other than the active site he proposed. So primarily majority of the multi-subunit protein or enzymes, they are basically allosteric protein enzymes, and they contain distinct regulatory sites and multiple functional sites. So in allosteric enzymes, such type of molecules which regulate the activity of allosteric enzymes are basically called the effector molecules. It can inhibit the reaction or it can activate the enzymatic catalysis or reaction. So the binding of effector molecule at allosteric sites trigger conformational change in allosteric enzymes, and that are transmitted to the active site. So the structural change brings in such a way that substrate can now bind or substrate can not bind to the active site. So these effector molecules bring this kind of structural change so that it can bind or not bind, and the reaction can be regulated in that manner. So this action at a distance through binding of one ligand affecting the binding of another ligand is basically the essence of allosteric concept. So when depending on the effector molecule, like basically the binding of effector molecule bringing what kind of change to the enzyme, it depends, like it can be positive effector molecule or it can be negative effector molecule. So if the binding of effector molecule facilitates the binding of substrate molecule at active site, then it is called positive effector molecule. Like if you can see in this reaction, what is basically happening, that these saffron color effector molecule, they are positive effector molecules. When they bind on the on the allosteric site, it is changing the conformation of active site, where E is written in the enzyme. So the conformation of active site is changed, and now this changed conformation of active site is very facilitative for substrate binding. So enzyme substrate complex formation can take place very nicely just by the binding of this effector molecule. So this effector molecule is called positive effector molecule because it is facilitating the binding of substrate molecule on the enzyme. However, if the effector molecule, when it binds and it changes the active site conformation in such a way that the substrate cannot bind to the active site, then in that case this is called negative effector molecule. So the binding of effector molecule brings such a change in the active site that the affinity of active site to the substrate molecule reduces, and because of this reduction in the affinity, the substrate molecule cannot bind to the enzymes. So so such type of molecules which inhibits the binding of substrate molecule at active site are called basically negative effector molecules. So allosteric control are basically information, all the allosteric enzymes are basically information transducers, and their activity can be modified in response to signal molecules. Then the allosteric enzymes play a very crucial role in many fundamental biological processes, and these processes are like metabolic regulation and cell signaling. Allosteric enzymes are also very unique as compared to other enzymes because its ability to adopt various different conditions, and they perform their regulation make them such a versatile that they can easily adopt various conditions. For example, like 2,3-bisphosphoglycerate is a negative effector molecule. So whenever there is strong binding of oxygen takes place in hemoglobin, what basically happens, like 2,3-bisphosphoglycerate, which is a byproduct of glycolysis pathway, a shunt of glycolysis pathway, produces, and then it binds to the hemoglobin, and then it reduces the affinity of oxygen to the hemoglobin. So basically oxygen gets released in tissues because of the presence of 2,3-bisphosphoglycerate, which acts as a negative effector molecule of hemoglobin. And then it is not necessary that all the allosteric enzymes will always be multimeric molecules. Many times what happens, like in case of, for example, glucokinase is a monomeric molecule, but its binding with glucose is such a the value, the Km value is so high that its binding with glucose is such a low that it shows a sigmoidal curve. So it is not necessary that all the time the allosteric molecule must be a multimeric molecules. Feedback inhibition. So feedback inhibition is also a property of allosteric enzymes. It basically occurs in allosteric enzyme. What happens in feedback inhibition when the end product of any biochemical pathway inhibits the regulatory allosteric enzymes. So suppose that, like, let's say glycolysis. Glycolysis is a pathway of almost 10 enzymes. So if the pyruvate or the end product, any end product, when it starts accumulating, then it inhibits some of the some of those enzymes, allosteric enzymes, which are regulated, which are regulatory allosteric enzymes. So it can inhibit those enzymes, and then the reaction can be stopped. So for example, as shown in this model, the X is getting converted to Z through Y. So if the concentration of Z is very high, or it starts getting accumulated, this Z can inhibit the enzyme E1 and can stop the conversion of X, or it can inhibit or stop the conversion of X into Y. So in this way, this feedback inhibition, or the over production of Z, or the accumulation of Z, can inhibit some of the regulatory enzymes, and then it can inhibit the pathway, it can regulate the pathway in such a way. So as we discussed, the increase in the concentration of Z means the cell does not need that end product. So that is why it starts accumulating. So if cell does not need that, so it has to stop the biochemical pathway which is synthesizing continuously the Z, and it starts accumulating. So after the product has been utilized, or it is broken down, then its concentration will again go down, and then this feedback inhibition which occurred through Z, as we shown in this model. So if the Z will go down, if either it is being degraded or it is being utilized, so the concentration of Z will go down, and then it will relax the E1 molecule, and then the conversion from X to Y will again start. So cells use this feedback inhibition to regulate enzyme activity mainly in metabolic pathways, because many a times those metabolic reactions are essentially required, or many a times they are not required. So cells use this feedback inhibition, and these metabolic pathway these feedback inhibition is basically evolved in such a way that this uses the end product, or many a times some intermediate products as one of the some intermediates as one of the negative effector molecule. So metabolic reactions such as anabolic and catabolic processes, they must proceed according to the demand of the cell, and they must be regulated by some of their intermediates or some of their end products in such a way that the metabolism is finally balanced and regulated. So in various anabolic pathways also, like in the synthesis of protein, synthesis of nucleotides, what basically happens, like sometimes in the synthesis of nucleic acid, sometimes the amino acids or nucleotides are basically acting as a feedback inhibitor. If suppose that the synthesis of protein is not required, in that case the amino acids itself start acting as the feedback inhibitor, so that more synthesis of protein cannot occur. And then the same thing is true with the nucleic acid or DNA synthesis also. Sometimes nucleotides itself acts as the negative effector molecule for the synthesis of nucleic acid. So and then it regulates the anabolic pathways. So, and majority of the time what happens, like these feedback inhibitors, they inhibit the first committed step of a particular biosynthetic pathway. So in this manner they can even save they can economically they can save even the synthesis of certain intermediates also. So if the first step of the pathway itself is inhibited, then there will be no synthesis will be taking place in the biochemical pathway. So a very highly studied model of this feedback inhibition is aspartate transcarbamylase by CTP. So at high CTP concentration, CTP binds to the allosteric site of very first enzyme, that is aspartate transcarbamylase, and it functions as allosteric inhibitor. So it decreases the enzymatic activity of the enzyme. So no further synthesis can take place, no further this biochemical pathway cannot further proceed. So then ATP function as a enzyme activator or enhancer in this biochemical pathway. So even with high concentration of CTP, sometimes the enzyme activity could be enhanced because of high ATP concentrations, which also acts on allosteric site in this enzyme molecule. So this example explains the benefit of allosteric control and the ability of allosteric enzyme to adopt in various different environmental conditions where the enzyme has to function differently, regulate differently. So the kinetic behavior of allosteric enzymes. So these enzymes consist of multiple subunits, and they are basically they are the enzymes of multiple subunits. So they are multiple active sites. They have multiple active sites per polypeptide chain, or one site per chain. So a chain with binding ability of the substrate at different binding sites. So in allosteric enzymes, the binding of substrate to one active site can affect the properties of other active sites in the same enzyme molecule. So due to this reason, the binding of substrate become cooperative, because one binding of one molecule is facilitating the binding of other molecule. So this phenomena becomes cooperative, and this effect of binding of one molecule facilitating other molecule can be of two types. Like if it is facilitating, then it becomes positive, and if it is not facilitating or it is inhibiting, then it occurs in negative manner. So allosteric enzymes or cooperative enzymes, they slow different kinetic behavior as compared to Michaelis-Menten enzymes. So those enzymes which have single active site, they follow Michaelis-Menten model. However, these enzymes which can bind allosteric effector molecules also, which can bind substrate molecule also, they generally show a sigmoidal curve. So sigmoidal curve, when the curve is plotted between the reaction velocity as well as the substrate concentration. So here the enzyme kinetics is basically the cooperative. So the increase in substrate concentration can facilitate the binding of more and more substrate on the active site. So the kinetics of feedback inhibition can be of different type. It can be of competitive, it can be non-competitive, or it can be partially competitive or mixed. So what basically happens in competitive inhibition, that the substrate, the inhibitor molecule itself is competing with the substrate molecules. They both bind on the same active site. So if the inhibitor molecule is bound, then there is no place for substrate to bind. So this is called competitive inhibition. And then in non-competitive inhibition, the non-competitive inhibitor bind on allosteric site, and what it does, that it brings such a change in the active site that the substrate molecule cannot bind. So that is called non-competitive inhibition. That means the substrate, the inhibitor molecule is not competing with the substrate molecule to bind on the active site. And when the graph is plotted in all these cases between Vmax or between the reaction velocity or the substrate concentration, the competitive inhibitors show the same Vmax, but the Km value will be different, because the substrate and inhibitor can have different affinity for on the active site. However, in case of non-competitive inhibitor, the Km will be same, but the Vmax will be different in that case. Allosteric interactions is basically defined as that when it occurs when the binding of a ligand to its site on a protein or receptor molecule is able to modify the binding of another ligand to a topographically distinct site on the same receptor, or vice versa. So in case of G-coupled receptor, which is given as example, one can see that the binding of the ligand on extracellular domain brings such a structural change in the intracellular domain that intracellular domain now can bind to some other substrate and can start the signaling events. And sometimes the binding at the extracellular domain can inhibit the binding with certain substrates, and then it can prevent the signaling. So this allosteric interaction through allosteric interaction, which facilitates the binding of ligands on the substrate molecule, on the receptor molecule, can bring cell signaling changes. To summarize what we have learned in this module, so the activity of proteins, including enzymes, often must be regulated so that they function at the proper time and place. So the control of enzyme activity is essential in maintaining the steady state of all organisms. So the substrate for most enzymes are usually present at a concentration close to Km, and that is why the substrate concentration plays an important role in regulation of enzyme activity. The enzymatic reactions are predominantly regulated by the concentration of substrate, enzyme, and then enzyme turnover, like the degradation and synthesis of enzymes. So different forms of enzymes called isozymes, they also regulate the enzymatic activity sometimes in different type of tissues or different phases of the life, like in the fetal hemoglobin has different affinity than hemoglobin. So beside this, the covalent modifications like post-translation modification, protein phosphorylation and dephosphorylation is the major regulator of enzyme regulation by covalent modification. So there are several ways by which the protein is being regulated, the enzyme activity is being regulated. So then beside this, the selective proteolysis of catalytically inactive proenzymes is also one way by which the cell regulates the enzymatic activity of the proteolytic enzymes, which are essentially required. So then the majority of the metabolic pathway, they depend mainly on allosteric regulation of the enzymes for regulating the metabolic pathways in the cells. So this is what we learn in this module. Thank you.